Next, we will discuss the process of testing the serum. Many veterinary hospitals are equipped with chemistry machines in-house to analyze the serum. Several commercial blood chemistry units are available. Some examples include VetTest 8008 by IDEXX, Abaxis' VetScan, Heska's iStat, and Hemogen's Analyst, among others. These machines are all easy to use and produce quick results for over a dozen blood chemistry values. In general, these units will complete a test within 5 to 10 minutes, depending on the machine. For further information, refer to your individual unit's instruction manual. Where such analyzers are not being used, the veterinarian is able to use outside laboratories to generate such needed information. Such tests are often combined into useful groupings we call profiles. A geriatric profile, for example, might test the specific chemistries that the veterinarian might suspect to be going astray in an older patient. A general health profile might evaluate a wide variety of chemistries that might help confirm early disease problems when a more general assessment is indicated. Filling out the paperwork properly is an important step if the laboratory is to run the correct test. A copy of the lab request form is generally retained for hospital records. This is especially important when the results are to be called in over the phone. Most forms are pre-printed on the back to accommodate the writing down of results. Accurate results also requires accurate preparation of the blood. When submitting serum samples, always make sure the serum is separated into another red top tube without obvious blood cells present. Or use a red top separator tube that isolates the serum from the cells. Separating the cells helps to ensure the chemistry values won't be altered by the presence of living cells. This is especially true as the blood cells consume glucose, possibly decreasing the actual values. Samples should also be kept cool so heat does not interfere with accurate results. Samples should be refrigerated during the day and placed in the pickup box with an ice pack in hot weather. Such precautions will assure the values are as accurate as humanly possible. Do not place blood and cytology smears in the same plastic bag with bottles containing formaldehyde, as this will often ruin the submitted slides. There are hundreds of specific tests and profiles that are run by laboratories to test for specific diseases and conditions. Some of these specific tests you may be asked to perform in your hospital lab as well. A few common examples of these are the feline leukemia, feline AIDS, and the canine occult heartworm tests. We will demonstrate briefly how one such test might be run. Because of the vast diversity of tests and manufacturers, it is necessary to redirect you to the manufacturer's directions for the step-by-step instructions. The occult heartworm snap test by IDEXX will be outlined here. Place 2 drops of serum or whole blood, as is being used here, into the sample tube. Add 5 drops of the blue conjugate and mix. Pour this solution into the sample well and wait for the solution to disseminate up to the activate circle. This will generally occur within 2 minutes. Once this has occurred, firmly press the button on the test kit until it securely clicks down. Within the next 6 minutes, 1 or more blue dots should appear in the result window. The single blue dot in this location indicates the dog does not have heartworm disease. If other dots appear, they should be interpreted as indicated on the instruction booklet. Some of these heartworm kits now include combination tests for conditions such as Lyme disease and Ehrlichia. These step-by-step kits are common and can be mastered easily. Another type of test used to evaluate for the presence of heartworms in dogs is called the heartworm filtration concentration test. This test looks for the presence of developing heartworms, called microfilaria. This is accomplished by first lysing or bursting the red blood cells in 1 cc of blood by adding a lysing solution to the blood. With the blood cells lysed, we can now push the solution through a tiny filter enclosed in this filter holder. This filter will trap any microfilaria present. The filter is carefully removed and placed onto a microscope slide. A drop of special stain is added to make the microfilaria show up more readily, and then a cover slip is added. With the microscope, the microfilaria's presence may be observed if present. A drop of blood can also be examined directly with a microscope for the presence of live, wiggling microfilaria, but the filtration concentration test allows us to concentrate a much larger sample of blood, making the test more sensitive. Many hospitals will use one or a combination of these diagnostic tests to evaluate patients for the presence of heartworms. We will now move to another method of gaining diagnostic information, the analysis of a urine specimen or urinalysis. A urinalysis may provide valuable information indicating a number of diseases: kidney disease, diabetes, and urinary tract infections, just to name a few. Collecting a urine specimen from an animal requires a bit of ingenuity. There are primarily three methods used for collection: free catch, catheterization, and cystocentesis. Let's briefly examine each of these methods. Free catch involves collecting urine during the animal's normal urination routine. This often involves quickly and quietly slipping a clean collection cup or bowl into the stream of urine while the dog is allowed to go out for a brief walk. A ladle or cup attached to a coat hanger often makes it easier for the collection of urine to go unnoticed by the dog. In order to get the most accurate sample, the urine should be taken midstream or during the middle portion of urination. Cat urine samples can be collected by placing Saran wrap over the litter in the litter box. The urine will pool in low spots and can be collected with a syringe. A clean cage without papers or other absorbent materials also works quite well for collecting a urine sample. Again, a syringe works well for collecting the urine off the cage floor. Using a catheter to collect the sample requires a bit more training, but will provide a urine sample with fewer contaminants. Some animals will be resistant to this procedure while awake, but this is an excellent technique for the anesthetized or very sick patient. The final method of urine collection is called cystocentesis. This method involves inserting a small needle through the abdominal wall and into the urinary bladder and drawing out a sterile, uncontaminated sample. Though this technique may seem to be a painful method of getting a urine sample, properly performed by the veterinarian, it can be done with little discomfort to the animal. The technique does require the bladder to be filled adequately so that it can be stabilized through the abdominal wall. The cystocentesis provides the cleanest urine sample of the three techniques discussed. Once a urine sample has been collected, it is now time to assess the components of the sample. There are generally three stages to every urinalysis: the dipstick, the specific gravity, and the microscopic sediment. Let's begin with the dipstick. Special chemically reactive test sites are located along the narrow strip. When urine is placed onto these sites, as demonstrated here, each site will react to the presence of whatever is being tested for and change colors during a specific amount of time. The results are read by comparing the colors to the standardized colors on the bottle. Each site should be systematically read and recorded in the medical records or onto a special lab form for the doctor to interpret. For example, the second site is testing for the presence of glucose. The color of the strip tells us that glucose is not present in the urine sample. The next item we generally test for is the urine's specific gravity. The specific gravity is a measure of how dense or heavy the urine is as compared to water. The instrument that we use to test the specific gravity is called a refractometer. By placing a drop or two of urine onto the window and allowing the free-floating window to lay naturally on top of it, a line will form on the scale inside, and the exact specific gravity can be read and recorded. Be sure to clean the windows with distilled water and non-scratching tissues before returning it to its case. The final step in a complete urinalysis involves preparing a sample for the doctor or technician to evaluate for a microscopic sediment. In order to separate the sediment, you must first centrifuge several cc's of urine for 10 minutes. The heavier components of the urine sample will be driven to the bottom of the tube and form what is called the pellet. The remaining urine can be discarded at this time. A drop of seti stain is added to the pellet and mixed by lightly flicking the bottom of the tube. The sediment is placed onto a microscope slide and covered with a cover slip. It is now ready to be examined under the microscope for tissue cells, blood cells, bacteria, and other items that might indicate disease.